It's really fun to be here, and I'm thankful for the invitations. It should be fun today. I think maybe I'll start with a digression that maybe opens. So I like to be totally interactive. So if that's okay with the teacher, we just ask me questions and stop me, and it's a long talk. So if we can ask questions, that's fun. It's interesting to think about this potential collaboration in using machine vision, computer vision, and biology. And I think that there's an explosion of cheap slow motion video now. High-speed video cameras are cheap now, and that's, I think, going to revolutionize biology because you can see things you couldn't see before because they were too fast. But it relies on asking good questions, and it relies on getting meaning out of these terabytes of data. And biologists don't necessarily know how to do that. And where it gets tricky is the synergy, because from experience I can tell you that relationships between people in different disciplines are tricky. If having a positive outcome where everyone wins is what you want, but it doesn't always happen, right? So I like to think of this swirly thing, right? So I can use this, right? So if you want the dream goal might be this, right? You have some mixing area, and in comes biology with some questions and some knowledge of what will change the field of biology. In comes computer science. We can do things that you otherwise couldn't, if you bring in some advanced techniques here. And then maybe this is math. And the ideal thing is, right, you mix these things together, and everyone comes back to their field with something that allows them to blow away their competition, right? So speaking as someone in one field, what would be ideal is that you come in here as, say, the biologist. You do stuff other biologists can't, and so you get all the grants, right? And this sounds cynical, but it's, that's the game, right? But this is tricky, and it depends a lot on your question. So I think you want to think a little bit deeply about these things. And I'm laying all this out here because I think that collaboration in computer vision with biology is a no-brainer. People like Lars are going to tell you today about ways that insects might inspire good computations and good algorithms. But sometimes that might not be true, right? So what I'm going to talk about today involves one of the good synergies, which I think is going from legged animals to legged robots, right? Our legged robots are getting frighteningly good, and I'm pretty soon I'm worried that biology may be irrelevant, but that's okay, right? But they weren't a while ago, and a lot of good robots were made by looking at animals. And the trick was to look for principles, not necessarily just mimic what the animals do, right? Take the principle and then use the best engineering techniques to get at it. But then there's other areas where, I've always been quite skeptical. My former advisor thought that some of the Roach data led to a new branch of mathematics. And I'll show you that slide and talk to you about that study. And I used to be very cynical. It's like that's just Bob being grandiose. Like, the cockroach cannon experiment did not found hybrid dynamical systems theory. But then I talked to some mathematicians last year. They're like, actually, that may have had a chunk of it to do with it. So I just thought I'd lay a bit of context and say that sometimes, these things aren't always clear winners, right? So maybe trying to design GPS inertial sensor fusion algorithms based on how pigeons think is not going to work. And it's better to take a standard engineering approach and use a big Kalman filter and it'll work. But then there are other areas where it's clear that our technology can't do things, and maybe there are animals that can do it and you can translate more cleanly. Oh, okay. Can no one hear anything? Can you hear me?
Yeah.
Okay. Oh, I'm being recorded. Oh, well, hopefully that was...
Stay closer around.
Okay, sorry. Hopefully that was unwise waffle, and so luckily it wasn't recorded for posterity. All right, so let's get on to what I'm going to talk about today. I'm going to talk about locomotion. So I'm interested in how animals move and how their nervous systems and mechanical systems work to achieve that and work together to do that. I'm from the Structure and Motion Lab, where we study lots of things, as exemplified by our crest. So this is our crest. We have a de-skinned horse, of course, so you can see the musculoskeletal system. We have an ostrich. We have force length curves. We have a cheetah and a GPS satellite, because these are all important things that you should have on a modern crest. So animals can do amazing things. This is a 500-kilogram mammal going 12 to 13 meters per second. It's going to jump over a 1.5 to 2 meter fence with an annoying primate on its back. Robustness is very good compared to robots, but not perfect. They get about 200,000 strides before they get a fracture and they get put down. But we'd like to know how they do this. What are the mechanisms that allow them to perform these amazing feats? One of the nice things about animals is that evolution has carried out a lot of experiments for us. So that's an animal that's really good at going fast on mostly flat surfaces. But there are animals like this squirrel that are really good at 3D heterogeneous environments. So this is an animal that can handle a complex 3D world. It can go up, it can go left, right, it can go sideways. Their ankles can go 180 degrees around so that they can face down in the tree and still the toppling moment doesn't pull them off. We know surprisingly little about squirrel biomechanics, actually. So that's what we're interested in. It's figuring out the mechanisms that underlie these behavior. And video like these are becoming ubiquitous, right? You can get a camera that does this kind of video. Now you can get these Casio Exelims for 800 quid, right? That'll do 300 FPS. And that's pretty good. You can do a lot with that. But 5K or 10K gets you something, research level. So who cares? Why should you care about locomotion? Isn't this frilly, woolly, fun biology time? Well, locomotion is the signature behavior of the kingdom Animalia, right? There's a million species of animals, and all of them are motile at some point in their life history, right? So figuring out how animals move has a lot to do with their bodies and their brains. Locomotion has deep evolutionary roots. So we think that locomotion is the evolutionary pressure that led to nervous systems in the first place, right? So it may be useful to sense things if you can't act, but probably it was when you could move that nervous systems, really were brought into play. Locomotion gives you a nice tool to try to tease apart the nervous and musculoskeletal systems to explain their structure and function. It's a unifier of molecular and organismal biology, so there's this divide between people that study clear liquids, what I like to call clear liquid science, where you're pipetting things all day, which is great. You should do it. There's a lot of big careers to be had in that. Versus organismal biology, where you're studying intact animals doing stuff. Locomotion is a nice way to bridge that gap. And then there's teaching and science communication. So locomotion is great because you can see it. It's animals doing stuff. And of course, there's lots of applications. So there's neuromuscular disease in humans. You'd like to be able to do a better job of helping people with illness. There's, of course, prosthetic limbs. So this is Hugh Herr from MIT. Does everyone know Hugh Herr? So Hugh Herr is one of the leaders in prosthetic limbs. He lost both his legs in a mountain climbing accident when he was 15 and dedicated his life to learning physics and engineering so that he could make better prosthetic limbs because he felt that lumps of plastic weren't good enough. He can give amazing, powerful talks on this. And this is a model, I forget her name, but she's also a big advocate for prosthetics. And his ankles, so his latest generation ankles, have a microcontroller that simulates frog muscle. So the richest data set on how muscle behaves came from frogs. They used that into this microcontroller. So the algorithm, the feedback loop that produces torques in the robotic ankle as a function of angle is based on a nonlinear curve that's phenomenologically determined from frog muscle. And, it's probably trumped up a bit, but Hugh says the first time he puts it on patients and they walk across the room, they start smiling because it feels right and it uses less energy. And it's his contention that an engineer would not have discovered that, right? So if you were an engineer just designing that ankle, you probably would have said, we'll use a PD controller or we'll use something, which would make sense, right? First principles. But this weird nonlinear curve is what actual muscle is like, including human muscle. And so that's, I think, one of the great motivations for a lot of this research. And of course, we'd also like to make robots. So you'd like to make more capable robots. For me personally, I would like to think that they'll be used to go into nuclear reactors in dangerous places and help people. But of course, there are also military applications. And so there's ethics to think about there, which gets interesting. So isn't this all solved? This is Big Dog. Everyone's probably seen Big Dog. It's really good, actually. I've heard depressing statements from leading roboticists in the U.S. like industry is too far ahead. Academic robotics needs to catch up, which is a strong statement from senior professors at Penn, for example. But it's really good. So legged robots are getting really pretty good. Is this bio-inspired? Medium. There's not a whole lot in Big Dog, I think, that's bio-inspired. They're using so it's a trotting gait that looks like a trotting animal, but that's a pretty descriptive observation. I don't think there's any deep control architectures that are coming from biology there. But having said that, animals still have a lot to offer. So this is from Life. So this is an elephant shrew escaping a monitor lizard, and they're just great. bounds along, adapts its gait, goes over, sees things, jumps over them, no problem. Slipping, sliding, grip is not an issue. Decides when to put its paw down, or doesn't need to, can get the turn done. No foot contact, doesn't matter, cycles the leg through. Lizard's not bad, actually. They're pretty amazing. We'd like to know how they do that. So, that's the context. I have a lot of different things to talk about, so I'll just launch into it. We'll wrap up with the context for the challenge. So how do the neuromechanical systems work together to produce locomotion? Well, what we're interested in is trying to get a more systems level understanding of how locomotion happens. And to do that, we take an integrative approach. So we're interested in combining, the bulk of what we do is experimental work, right? So we have gliding animals in the wild, we have cockroaches running on condoms, we have dogs on trampolines, and we have measurements of the muscular system of cockroaches. And we try to do these experiments and fuse them together to get an overall picture of what's going on. We try to gain strength from integrating with mathematical and physical modeling. So we make models of the animal in systems of equations, and we do it as well with physical models like robots. So the nice thing about robots is if you think they move like your animal, you can do a lot of experiments that you couldn't in the animal. So for instance, you can control the brain of the robot, but you can't control the brain of a dog. So we can program the robot with the putative controller that we think the animal is using, and then it'll do it every time, and we can see what happens. There are also great ethical benefits to using robots, right? We'd like to kick the dog to see how it recovers from being a sudden lateral perturbation, right? But we can't get ethics to do that, and we don't want to do that. But you can kick the robot. Nobody cares. And a lot of the undergrads love to fix them. So, it's a win-win, right? And then there's instrumentation. So we're bringing in more and more micro-instrumentation. So we'll talk about some sensor backpacks and some nanotechnologies as well. So let's talk about this first topic, the neuromechanics of leg locomotion. So when I'm talking about neuromechanics, is that just a fancy word, or does it have some guts? The reason to add mechanics became clear a few years ago. So we know what stuff does locomotion, right? You have sense organs that send information up into your spinal cord and brain. Signals come back out that turn on muscles. These muscles pull on joints. Those move the body segments relative to each other. That pushes on the ground, and you move your body relative to the world. But we don't really know how that stuff achieves locomotion. We know pieces of it. We know what muscles do roughly in isolation. We know what neural reflexes do in isolation. We'd like to put a lot of that back together. And the classical view, this is a bit of a straw man, but going back not too long ago, the classical view of control was that you have this high-level control in the brain. You have things like oscillators in the spinal cord. You have the musculoskeletal system in the environment, and there's a lot of feedback coming up all the time. So if I'm going to grab my coffee, I'm using eyes and proprioceptors in my muscles to bring this arm in and grab the coffee cup. One problem with this is the so-called degrees of freedom problem. And that is that, even a cockroach has 1 million neurons, maybe a couple 100,000, 220 muscles, and 42 degrees of freedom. So what do they do to compute what to do with those 42 degrees of freedom in the next millisecond, right? How does the nervous system computationally handle this task of figuring out what to do in this huge space, right? Our computers can't do it in real time. So how does the nervous system do it? So people got interested in this problem and started looking at it in things like insects. One of the classic studies was cockroaches running on rough terrain. So this is a cockroach running over rough terrain. They blast across it at roughly their preferred speed. It doesn't slow them down much at all. If you measure the activity they're sending to their muscles, it's the same. So if you measure what spikes the nervous system is sending to a couple of the biggest muscles, it doesn't change. So they seem to be doing this without a lot of feedback, right? That wasn't good enough for Professor Full. He wanted more definitive proof that this was a mechanical stability, which was what they were hypothesizing that it was. And so they put a cannon on the back of the cockroach. So this is the famous cannon study. This is a cockroach running with a cannon on his back. And the cannon fires here. Cockroach is shoved sideways. And I would say it's recovering there, which is 12 milliseconds. And the fastest reflex arc in the cockroach is very fast. But to go from a joint sensor, a proprioceptor in a joint being perturbed, a spike to come into the nerve cord, even a monosynaptic pathway, a single synapse, and then a spike to go back out, and then a muscle to start developing force to counteract that, should take at least 30 milliseconds or more. So this recovery is not neural, right? It must be something else. And so they hypothesized that it was mechanical, or had to do with the dynamics of the animal. And so the story is that they took this to Phil Holmes at Princeton and said, Phil, will you model our cockroach? It's really interesting. And Phil said, no, it's a waste of my time. I'm much more important than this. They said, no, it's super cool. Please model it. And Phil said, no. Eventually, after several beers, they got Phil to agree to model the cockroach. And what they discovered was pretty amazing. So you can simplify the roach down. They run on tripods. So they have 6 legs, and they run on 3 legs, and 3 legs, and 3 legs, and 3 legs, alternating tripods. So they approximated each tripod as a spring, and the roach's body as a hockey puck, and they bounced it forward in a simulation, right? So they gave it a few parameters. It's called the lateral leg spring. It has 6 parameters. It's a piecewise energy conserving system, but it bounces into these legs that keep resetting. And these aren't quite the exact videos of that model, but they're close enough to give you an idea. They set the model going. They hit it with a cannon with purely feed-forward commands, and it can recover. So it suggests that if evolution were to tune the dynamics of the animal to behave like that spring hockey puck system, it will have the stability properties of that spring hockey puck system, which can respond to perturbations faster than a reflex. Right? So if evolution tunes the whole animal to emergently behave as this system, it will have those stability properties, and it can recover faster than it can think, which is super cool. But then if you want to tease apart how feedback is probably used, because the animal does use feedback, right? It just can overlay it over on longer time scales. So then you have to think about, well, given this hockey puck dynamics, if you're perturbed now and you sense it, when should you inject some impulses to stabilize the system in a way that doesn't destabilize it further? So it gets tricky. And that's why we add mechanical, right? You need to know the mechanics of the body in order to correctly interpret what the nervous system is doing. So this is the output of the model, and they found that just that simple model is, for example, neutrally stable in heading and velocity. So you give it the cannon, and its heading will change, its velocity might change, but those will go to new values, whereas the eigenvalues for orientation and rotational velocity are asymptotically stable. So you kick it, it'll wobble a bit, but then it'll come back to an orientation about the velocity direction, the vector that's stable, and the rotational velocity will be damped out. One cool thing they did then was they said, well, where is the cockroach in terms of this model, right? So if you ask the model the question, what is the most stable speed to move at, right? They get this curve. And the most stable point is here. So where are cockroaches? There, right? So then you ask, what about body mass? This is your curve. Animals are there, right? So this is the evidence that the model is, that animals choose to move in this dynamically stable region of the parameter space. So that was a cute little extra study. So this has led to this sexy term, preflexes, which some people hate and some people, it's basically passive dynamic stability. So if evolution tunes the musculoskeletal system, it can handle a lot of variation in the environment in a feed-forward manner, right? So that's really interesting, and it asks the question, then how is control executed, right? And so maybe what happens is the way that the body solves this degrees of freedom problem is through hierarchy. And so high up in the brain, actually, planning is done on a lower dimension space, which is a template of the body in some simpler sense. So just a single leg spring that approximates all 3 legs, and you need to know the angle that you're going to put it down at and the stiffness of that spring. And the brain plans in those coordinates and then sends that signal down to the nerve cord, and that's transformed and sent out to the legs. So that's where we're at now. Let's see what I want to say here. So giving that a bit more flesh, that's a very schematic diagram. This is what it would look like in, say, a cat, right? So the cat has a brain. It talks to the spinal cord. This turns on muscles. Those give the body some dynamics. You have sensory receptors that measure what's happening. You have exteroception that looks at the outside world. And all this comes back in. And the central problem that we deal with is teasing apart this stuff, which is neural, from this stuff, which is mechanical. When I get to a later point in the talk, I'll talk about some new technologies that will enable us to do this, I think, in much better ways. So, there's a lot of theory out there. So if you ask, what are our hypotheses for how the nervous system handles this problem, there are a lot of them. So there's the templates and anchors hypothesis, there's optimal feedback control theory, there's hierarchical optimal feedback control, there's the uncontrolled manifold hypothesis. There are loads of theories, actually, for this. Some of them even can make predictions that are quite general, if you have a good model of your animal. But I think the truth of the matter is, we need more data. And we need data around 4 questions that structure what I'll talk about from here. So I think the first thing we need is what are the targets of control? And what I mean by that is we need to know like what the animal does in normal steady state running or in its normal steady state behavior so that we know when it's recovered. Right? I'd like to know if these are general principles across size and morphology. That's more of a biology question, but I still think it helps you. By comparing animals, you learn a lot about each of the animals that you're looking at. So that still, I think, is very useful. You'd like to know about the consequences of having to tune your control architecture across behaviors. And finally, we want to know how sensory input is used in fast locomotion. But overall, we need data. We need engineering. We need to get the rich data sets that will answer these questions. So on this first question, moving to talk about legged locomotion. One of the coolest findings in biomechanics is that animals, no matter how big they are or how many legs they have, when they move at running speeds, they look like a pogo stick. So all animals at roughly running or trotting speeds group their legs together into sets of n/2 and then bounce on them. So a human just runs, boing, 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 and their forces look like this. You get a hump in the vertical direction. You slow down and then speed up because you hit the ground, load your leg like a spring, producing a retarding force that way, and then you unload it and you get that force, you get that work back. Dogs, 4 legs, group them together and trot on diagonal pairs. Cockroaches do it on tripods, so they have 6 legs, they take 3 at a time and they bounce on them. And then crabs have 8 legs and they take sets of 4 at a time and they bounce on them. Well, one of the interesting questions is how do you use this control architecture in the real world? And if you take a man from the 70s with a handlebar mustache and you run them over cushions, what you find is actually really interesting. So as the surface gets softer, goes this way, humans will stiffen that virtual pogo stick leg. And one of the first things I asked when I came to this field was, well, can many-legged runners do that? So if cockroaches have the same pogo stick behavior for their virtual leg made up of 3 legs, can they stiffen it? Right? And so this was really fun. So the first thing we needed was a pillow, a cushion that was scaled for a cockroach. And so we looked around and it turned out latex was the right material. So insert lots of jokes here about condoms and dental dams. But those turned out to be the right surfaces. So we ended up using sheets of latex to make this surface. Calibrated it with a force lever. I did this work with Shai Revson, who was a great scientist. And so we found some surfaces that had the right stiffness for cockroaches. And then we did some cool sensor fusion stuff. So it looks kind of ghetto here, actually, but this was, this is early on. The fish prototype's much better. So we wanted to measure the center of mass mechanics of the cockroach on the elastic surface. And normally you get these kind of mechanics from force plates. So, you have these strain gauges and some kind of plate where you can get the forces of the animal as it runs. We couldn't do that because we were going to have the animal on this soft surface. And so we ended up putting a 3-axis accelerometer that we could superglue to the back of the cockroach. And that was good, but the problem with accelerometers, as you know, is that without fusing with anything, it's very hard to integrate to get orientation nicely, especially over long times. And we only had an accelerometer. We had no gyros or no magnetometer, so we really couldn't have done it with just the accelerometer. But we decided to fuse the accelerometer data with the cross because we wanted the acceleration data so we could be close to force, and we also needed the orientation data so we could get the orientation of the body out. And so we built this 5-point cross with an accelerometer at the base of it, and then we had our setup like this. So we had a high-speed camera above. We had a nice setup where we could bring light down that came sort of in an epi-illumination setup, actually. And then we had an ABA design, so the animal runs from a hard surface to a soft surface to a hard surface. And then we got coding, and so we used an unscented Kalman filter to do a nice tracking of the cross. So what's happening here is we have a single camera that we've calibrated, but then we use the Kalman filter to fit a 3D model of the cross through the camera transformation and the rigid body transformation of the cross into the world. So that was really nice. So that basically what the Kalman filter spits out is the rigid body transformation of the cross in the world coordinate system, which is what we wanted. And it does it through the camera transformation. And then they also think the nice thing about it is we can use it to predict, to solve the matching problem. So we would use the Kalman filter to say, where will the cross be in the next frame using the system evolution matrix? And then we could figure out where these points would be in the next frame and then look for them in the movie. So it helped us both track the cross through the movie and then get out the 3D coordinates in the world frame that we wanted. So that was pretty cute. And I was amazed that the UKF could do this through these nonlinear transformations. Just maybe falling off a log there. One here, I see people are nodding off UKF, but the results were really cool. So the cockroach is running here on the hard surface. It's going to encounter the dental dam here. They blast across it, despite it being, it's really soft actually. It's softer than their virtual leg. But a big thing that you notice is their vertical accelerations become a lot smaller. And so that's different from the humans. Humans stiffen their legs. So in the human, you would expect this to be the same. And so we wanted to know what was going on. Are the cockroaches doing anything? And so we turned to a mathematical model, a refined version of that spring-loaded pendulum. This one has a torque at the hip, and it drives the leg around. And in that way, you can model the animal sinking down into the surface. So that was the idea. So we needed to refine that pogo stick model so that it models the legs flying through the air. And so that's what we got out of it, is this model of the roach. We're going to confront it with a simulated soft surface here, and it sinks down in. And what we capture with that is that the legs that are flying through the air can hit the ground sooner because it's sunk into the surface. And that turns out to be enough. That change in posture at touchdown turns out to be enough to get out the behavior that we saw in the animal. So we think the roach isn't stiffening its legs. It's sending out a leg with the same virtual leg stiffness. So that's really interesting. Maybe they are dumb after all. I like to say, and I'm sure Lars says, insects are really smart, and they're smarter than people give them credit for. There's a great paper that argues that insects are smarter than mammals. Basically, if you just count behaviors, they do a lot more stuff than, say, a cow, and so they're actually smarter, which I kind of like. I think that's really cool. But that's really interesting. And so, but of course, there are lots of alternate hypotheses to explain this, right? So cockroaches and humans are very different. It could be a lot of different things. It could be they're different size, they have different posture, they have different leg number, right? Rather than this constraint on neural control. I'm sort of hinting at, cockroaches can't do it, their brains can't figure out how to stiffen a virtual leg made up of 3 legs. But, there's a lot of other stuff involved in that. So one way we can sort of try to tease those apart is to look at other animals. And so then we ask the question, what does a multi-legged animal that still has to coordinate multiple legs into a virtual leg, but it's big and it's upright, so it might care more about falling over, right? And for that we chose a dog. And so this is the structure motion lab. This is our array of force plates, and we put a bunch of cushions that were scaled to the right stiffness for dogs. And those we calibrated there. We ran a bunch of dogs over these cushions, which is a lot of fun in a student project. And we tracked them using motion capture. So here now we're getting loads of 3D coordinates out of a mocap system, right? And what we found was really interesting. So as the surface gets softer, dogs don't stiffen their virtual leg. They're about the same. And if you remember, humans were doing this. So this is another interesting piece of the puzzle. So far, stiffening the virtual leg appears to be unique to humans, right? And so what we think is going on is that it probably has to do with face planting. Well, there's a couple things. So one is maybe humans can do anything, and in the human study on cushions, they kind of whispered into their ear, do what feels normal, whatever that is, stiffen your leg, because they thought that would be cooler. Or it's that humans do that because they don't want to face plant. If you're an upright 80-kilo biped and you're going fast, you can do a lot of damage to yourself if you fall over. Dogs are pretty close to the ground. They're still upright. They still can get hurt. They have a significant energy. Roaches probably don't care a whole lot. So it would be interesting to do some simulations to look at the stability as a function of stiffening and not stiffening your leg. But another way to tackle this question is to go from to use some technologies from engineering to try to answer this question. We talked earlier about you can't kick dogs, right? The other thing you can't do that easily is measure the energy consumption in dogs. You can do it, but it's pretty hard, right? And it's also hard to bolt on legs of different stiffness in dogs. So you'd like to ask a dog to run with stiff legs and then with soft legs, but that's hard. Maybe you could actually with like a dressage dog. So we had an EPSRC grant to do this, which was great fun. So working with Dan Kodacek at Penn, we were able to we actually funded the very first of the latest generation of the Rex robots. So you know these six-legged robots that have been semi-legendary. They're called the Rex robots. And so we bought one of those robots to ask questions about why insects and dogs don't stiffen their legs on soft surfaces. So we took Gary, as we affectionately call our Rex robot. So Gary is about 7 kilos. It's about this big. It's based on the cockroach. It has fiberglass or carbon fiber legs. And we ran Gary over loads of different surfaces that were scaled to be the same as what we had confronted the cockroach and the dog with. And the nice thing is it just tells you how much energy it used, right? So it logs voltage and current, and you can just compute watts used in the last few runs, right? You don't have to put a mask on the dog, which is great. And what we found is quite interesting. It looks like, basically, that every time we put stiffer legs on the robot, irrespective of our four surfaces, the energy consumption went up. So it suggests that it could have to do with energy for the multi-legged animals. So it's actually better for them to move with softer legs on these surfaces. And if they don't care about stability, then it makes sense. Okay. How's this going? Any questions? Interesting? Not interesting?
Interesting.
Okay, good. Computer vision? Not enough computer vision? Yeah, Ricardo.
Did you try to do the same experiments of the person running with a kid?
Oh, great question. Actually, people may have done that. I don't know of the experiment, but I would be surprised if the motor control or child development literature hadn't looked at that. Why?
Because they are shorter.
Yeah.
Than a normal person.
Yeah.
So they may care less to fall down. And
that's a great
they are probably willing to do that. Because
what's that?
They're crawling kids.
Well, crawling, I think. Yeah. No, you could do that as well. But so there's another way to get at that, and I would like to do that, which is to look at birds. So we have a lecturer in the lab. Monica Daly does this kind of work in bipeds in birds. So she has birds on treadmills with obstacles that come around every now and then. Her famous experiment was a trapdoor, tissue paper trapdoor. So a guinea fowl running along, and then there's a tissue paper that looks like floor, but it's not, and so they fall through it, and they don't know it's coming. Pretty cool experiments. But I think that's a great question. So we would like to know whether small bipeds that presumably care less about stability stiffen their leg. It's tricky because there's also changes in morphology. So small animals have much more zigzaggy legs. So small birds have these really sort of crouched posture, and so it's different because that is thought to give you a lot more stability, or at least range of motion that you can use for things. But yeah, no, that's a great question. Great. So this is evidence that maybe dogs don't increase their leg tips because it would cost more energy. And we did a lot of outreach with the robot. Robots are great for outreach. We went to the Science Museum. We didn't run over any kids with the robot, which was amazing. There was so much Wi-Fi that every now and then the robot would just go straight ahead, and you'd have to have wranglers that grabbed it before it ran over any children, and that did happen. So we had 4 days of outreach with like 8,000 visitors. Really fun. Ed Miliband came at 10 a.m. on the Sunday, which was really funny. So it was cool. So what about another important aspect of this, which is that you have to do lots of things with your body. So you have this control architecture, and what we've been doing so far has been perturbing the environment, right? Letting intact animals do all of this, and then measuring what happens. So, but we have no idea. We're black boxing a huge chunk of things, right? We're dialing in mechanical perturbations. We're measuring the mechanics of the animal, but we have no idea what's going on in the nervous system. So we had an idea to try to do an inside-out approach rather than this outside-in approach. So what if we could dial in changes in the neuromuscular system, have them play out through the body, and then see what happens? So dial in neural changes and look at mechanical changes. This was done with Simon Sponberg, who was a PhD student at the time, who was really, really talented. So we put in electrodes into a cockroach muscle where we could measure the spikes and then discriminate them in real time and then add in spikes. So we patched in real time to the animal. So we would pick up a couple spikes, discriminate them, put in a delay till the next step, and then add spikes onto the next muscle action potential. So in the next step, the two normal spikes would happen, and then we would overwrite this one and then add extra ones. So we could make this muscle flex a lot more in the next step. And the nice thing about that is it happens in the context of the intact, freely behaving animal. So however we modify these neural commands, they're played out through the physics of the body. Right? We used our backpack. And this is highly relevant to today. So to get enough data for this question, we had to develop some techniques to track the limbs because we wanted to know the kinematics of the limbs. And so we developed some software that would cut the cockroach out, put it into body coordinates, and then track the tips of the tarsi. So these kind of algorithms are more and more important. So the two aspects of this are really important in locomotion. One is the real-time aspect of it. So doing this real-time measurement of the animal's state, and then applying something based on its current state is really big and going to get much bigger. And second is having reams and reams of data that you can automatically process in this manner with algorithms like this. So what you're getting out here, these are the x coordinates of the tips of the tarsi, right? So these oscillating waves here are the 6 legs, so it's the x coordinate of this tarsi tip as it goes forward back through these cycles. There's the one for each leg, and what you see is that you've got 3 legs that are in phase, and then the other 3 legs that are 180 degrees out, and that's the 2 tripods that you can immediately see, right? And this sets the context for the challenge somewhat, because analyzing these kind of data are really useful for trying to understand the neural networks that generate gait, right? And that has applications of course, in medicine with people who have gait abnormalities or detecting gait abnormalities, and in basic neuroscience. Roaches are hard because this, if you look at just the phase of these oscillators, you're talking about a 6-dimensional torus, which is hard to visualize. And so people have worked on trying to model these controllers, but it's hard in insects because it's a 6-dimensional torus. In the mouse, which we're going to talk about later, it's a 4-dimensional torus, which is tough, but you can take phase differences to get them down to a 3-dimensional torus, and then you can see it, and things get really exciting. So we'll talk about that. So coming back to this experiment, though, if the cockroach is standing still and you patch in spikes, you get a big twitch. It's a middle leg that we're turning on an extensor muscle. And if you add 1, 3, 5, and 7 spikes, you get actually pretty linear responses in pitch, yaw, in the pitch and yaw variables as you do that. If the animal is running at its preferred speed, and you patch in up to 7 spikes in here, I defy you to tell me at what frame in this movie that happened. So in the middle of this movie, that extensor muscle, which is a big extensor muscle, is being given 7 spikes, which is the most we ever saw naturally in the running animal, right? So this tells you how important context is, right? The animal has enough, the limb and the animal have enough inertia and enough co-contraction of other muscles in the limb that the context is critical. And when you're running fast, things change dramatically. It seems kind of like a no shit, but decades of anatomy have said this is an extensor muscle. But actually, in a fast-running animal, it doesn't extend anything, right? So things are very complicated, and you need to take into account the context to figure out what things actually do. It's a little bit cheating because the animal probably would never send a wave of normal activation to all of its muscles and then 7 spikes to muscle 142, which is the condition we've created, right? What we'd like to do is get higher up and tease apart the waves of changes that are sent to do things like turning, etc. But we did find that if you advance the phase of spikes, so rather than patching over and making a bigger muscle contraction, you advance the phase of it, you can turn the animal. So what happens is the leg doesn't go as far forward during swing, it hits the ground sooner, and the animal turns. So we could reliably turn the animal. I wasn't comfortable with submitting big DARPA grants based on this to do remote control insects, but other people might. So yeah, you can turn the animal. So muscles are multifunctional, right? And the nervous control is highly state dependent. And that's why we need these real-time techniques, right? So the more that you can measure the state of the animal in real time, the more you can gather enough data to tease apart this context-dependent nature of the nervous system. So we would love to have, if you have real-time pose estimation of these animals, then you can test hypotheses of how the controller behaves in that when the animal's in that state, right? And you have a chance to trigger perturbations that tease apart that controller when it's in that state only. So you can get enough data to do these things. All right, cool. What am I doing? Do you want me to go to 11?
Yeah.
Okay, great. Is everyone okay? Still awake? Stretch your legs. Keep going. All right. So this is interesting. You guys may not have seen this topic, but I thought I'd throw it in there because it has different sensors. So this is using backpacks. So one of the other questions I'm interested in is gliding in mammals. So we've done some nice work looking at these guys. These are colugos. They are flying lemurs that live in the jungles of Singapore. And it's a misnomer because they don't fly and they're not lemurs. So actually they're primates. They're really closely related to us and they glide. But there you have it. There is a female with a baby underneath. They're nocturnal. They'll glide 50, 150 meters in the night from one big tree to another and then climb up the next tree. And we don't really know how this evolved, why this evolved. It's an interesting question. And it could have really good explanatory power on the question of how flapping of flight evolved in mammals. So does anyone know the one mammalian species that has flapping flight?
Bats.
Bats, yeah. So that's the only time flapping flight has evolved in mammals is bats. Good. Didn't get called out on that one by anyone. So, but gliding has evolved lots of times in lots of different, in all the major lineages, right? So, going into fish, right? So it seems to be it can be a very useful adaptation, and some of the reasons are thought to be that it evolves because of the habitat structure to get away from predators, avoiding injury. There's a few different reasons why you might want to glide, but it's hard to get at, especially in these colugos that are nocturnal. So we had the same hypotheses for colugos, but we had no data until we put together some backpacks. So this is really cool. This is when accelerometers just came out, and they were like really exciting. So we built this in 2004, right? And we did it ourselves, and you can see we're like not proper double E's. Look at our traces are all not like, we didn't even do like 90-degree traces, and it's like super ghetto, but it worked. We glued a couple accelerometers on there. We have an NAND flash chip and a microcontroller, about yay big. Would run for 80 hours, logging 3-axis acceleration at 100 hertz. Right, and Greg went out to the Singapore Zoo, and the way you catch these animals is you go out with your partner, and they're sleeping in the mornings, and your partner spots one and says, "There's one on the other side of the tree there," and you sneak up from the other side of the tree like this. The animal's on the other side of the tree right there, and then you go, whoom, and you grab it and you pull it down, and they're big and they have claws. They're a kilogram, and when they unfold their wings, they're like the size of a doormat. So I don't envy Greg doing this. But then you shave off some fur, and you super glue on a piece of wetsuit, and then the data logger on top of the wetsuit, and they glide fine with it. It's much less than the weight of their babies that they glide with. And after 2 weeks, the super glue fails and it falls to the ground. You pick it up with a radio tracking beacon, and you download the data. It's sweet. Now you can go nuts, and we want to go nuts, right? You could have GPS on there. You could turn it on and off when the sensors pick up movement. Like, this was super crude, and you could do much, much better now. And people are, so it's exciting. So we had this massive sample. So we got this onto 6 animals, and these are what the data look like. So that's climbing. They're really good climbers. They can bound up trees really quickly like that, using their claws and gripping the tree trunk. And the data from the accelerometer were great. You can see these behavior nicely. And this is another area that's an interesting question. Is I think Aldo Faisal at Imperial is doing nice work with sort of behavioromics idea of automatically classifying behaviors and doing more fancy informatics with it. But nice, easy to use generalized algorithms that annotate these data would be great. So you train it with what climbing looks like, what gliding looks like, and then you give it several gigabytes of data, and it spits out the data. Would with probabilities on it would be great. And I don't think that's a super hard problem, but it's not one that, you know, the biology community isn't necessarily aware of it or doesn't have it. So that there's a good link to be made there. But as per my swirly diagram, it may be a really boring problem for computer scientists. So you want to hammer out at the beginning of the collaboration what everyone's going to get out of it and whether it's going to be interesting or useful for the CS side of things. And then gliding looks like this. This is pretty amazing. So these guys. Do that. That's slowed, and then in real time. Looks like that. And one of the things you notice there is that right before landing, it looked like the tail was flapping. And so we don't know whether that's passive or active. It looks active. If it's active, it could be a big link in the evolution of flapping flight in mammals. And we'll be sending that to nature soon. Cross your fingers. So this is what a glide looks like. They're on the tree trunk. They push off. They glide. You get some dynamics and acceleration, and then they land. And what you get out of this is really unprecedented. So this is 6 days in the life of one of these animals that you can't really see, right? So these technologies allow you to do natural history on animals that you can't see, right? And that's huge. Typically in the past, people would see a colugo in the tree in the dusk, and they'd say, there it is, and then it would glide away. And they'd be like, it glided. And then they go home. You know, so this is great, right? We can record what it does every moment for 6 days. And answer lots of interesting questions. So they're mammals and they behave like mammals. So females tend to glide, climb up the tree, and then eat. And then sometimes they'll glide again. Males will glide, climb up the tree, and then often glide again, as opposed to forage. And so it suggests that, like a lot of mammals, males are doing territory defense or mate searching, as well as foraging, where the females are mostly foraging. You can't infer anything about how humans should behave from this. This is an animal, it's different. We all know about the naturalistic fallacy, right? You can't apply it. Okay. So we've done some other work looking at the systematics of this. I'll skip over this because it's less important. It looks like we can ask questions about whether it's really cheap to glide, and actually it doesn't look like it. It costs so much to climb the tree after you glide that actually gliding isn't that economical compared to going quadrupedally through the trees, which is really funny. If you ever see them moving on 4 legs, it's bad. They can climb, they can glide, beautiful. Put them on the ground, it's not pretty. So that's interesting. People thought it was about economy. Oh, you're gliding, it doesn't cost anything, it's super easy, but you've got to climb the tree when you land, right? And then there's injury avoidance. So interestingly, the longer they glide, the less the impact force. So getting set up to be able to parachute is important. And this is interesting to compare with ballistic leapers. There's a lot of animals that just leap and they can't parachute, right? So they're limited in what they can do. And then there's a big section that's probably about control. So this is a North American squirrel that does a bad job of gauging where this pole is, and the center of the pole is there, and they hit the left side and get bonked off the edge a bit. So you want to have some ability to modulate your aerodynamic forces through your landing. Unfortunately, the beginning of that video doesn't play well, but squirrel goes bang, and it's fine. So, all right. So that was colugos. And next thing we want to do is do some modeling. So we made that whole backpack, and then we went to our friends and said, we made this backpack for flying lemurs, and then I sent it to my friend Doug, and he said, well, why didn't you just buy this one? So that was annoying, because we spent ages making it. The next thing we want to do is we're looking at deforestation. We want to look at new backpacks to get at GPS out, and then climbing and gliding robots. This is actually really interesting. So now going the other way, thinking about how biology might inform engineering, we'd like to make robots that can climb and glide. And so Jonathan Clark at FSU is working on some of this. And he's got this really poor approximation of a colugo, right? It's an airplane with legs. It's not he would kill me if he knew I was saying this. But, this is the way we have to do it with our current technologies, right? It's a glider that has legs. And it's neither a very good climber nor a very good glider, right? So I think the inspiration there is going to be materials, isn't it? It's coming up with these extendable, flexible wings that are lightweight and can do what you need. So that's going to be really fun. That's our next big challenge there on the technology side. All right. So now I'm going to come back to do more leggy stuff. Let's talk about this wacky big idea that we're working on now. So when I talked about the roach where we turn on the muscle, we had a pretty bad handle on it. Remember I was saying that this is pretty artificial to turn on one muscle out of all of them. We'd really like a better way to perturb the nervous system. And there are multi-electrodes. You can do it sort of electrically. But there's a revolution happening in neuroscience, which is this new technique called optogenetics, where you have much better control over the nervous system. And we're going to start using that. And this is why mice are really worth looking at now. So we'd like this. We'd like to be able to dial in mechanical perturbations and measure them. We'd like to be able to dial in precisely the neural perturbations. We'd like to have the wonderful freedom of cellular and molecular biology where they can do lots of genetic manipulations, apply lots of drugs. We'd like to be able to perturb the mechanics. We'd like to inject noise here. We'd like to switch on and off sensory feedback. A lot of what's been learned in motor control has come from horrible things like crippling cats by cutting nerves in their ankles and stuff. In the 1930s. We learned a lot. It's a really tough ethical question. If you take a cat and you cut the connection between the brain and the spinal cord, you hold the cat's body up and put it on a treadmill, the legs will go through gait transitions from walk, trot to gallop. Right? So the higher centers aren't needed. Just the spinal cord and the legs and the muscles will walk on the treadmill. It can't hold itself up. The balance isn't there, but the patterns for generating the different gaits are there. Was it worth doing those experiments on those cats to learn that? It's tough. It's probably, Christopher Reeve before he died would have said yes. And he was a strong advocate of research. I went to a talk where he said, I wish I was a rat, because then experimental procedures would be allowed to be tried on me. But it's not permitted. Right? So, but I think what's amazing is that we're going to be able to do these things also much more ethically because of these new neurotechnologies. So we'd like to have much better handles on all these things. So the idea is to sort of set up a systems biology type approach to neuromechanics. So in the same way that clear liquid scientists talk about cells and molecules and different pathways in this sort of dynamic sense of how they all work, we'd like to do that with muscles and skeletons. And there's an English professor, Sir Dennis Noble, who is a big champion of systems approaches. And he offers these 10 principles of systems biology, which basically, if you boil it down, is be open-minded. It basically means don't be necessarily reductionist. You might learn something by thinking about higher levels. It's not always brilliant to just drill down to the bottom. He's written a whole book on it, which is interesting. And he looks a bit like James Bond, I think, which is cool. But I also like Wikipedia's definition. Systems biology has the ability to obtain, integrate, and analyze complex data sets from multiple experimental sources using interdisciplinary tools. That's not bad, actually, for what we want to do, right? We're going to have video data, we're going to have muscle recordings, we're going to have different experiments we do, and then we're going to take advantage of all the strengths of different disciplines. So the neuromechanics community does get this. And we're getting there. We have integrative approaches across length scales, disciplines. Theory and experiment we're not so great at. Biologists are afraid of math. We seek these good models of biological processes. We do have some different data sets, but they're not amazing. Genelia Farm has a thing called the Fly Olympiad, where they're creating like the Olympics for flies, and they're going to make flies go through all these challenges and then try to integrate those data into like what's going on with their genes. So like this fly couldn't jump over this gap, and it's because gene X is bad, which is a really interesting informatics challenge, actually. So the three areas we're pushing this with our grant is on the neuromechanical and theoretical sides. We're trying to center things on mice and make a lot of progress to set up this systems approach. On the neural side, we're developing optogenetics techniques around neuromechanics questions. So optogenetics is a new field where you can genetically modify animals to express light-sensitive proteins in certain cells, and then when you shine light on those cells, you can switch them on or off. So that's the way it works. So nerds who loved pond scum, biologists who love pond scum, were studying archaebacteria and discovered a nice template for these channels and sequenced it. And that's what allowed us to engineer them and put them in other animals. So it's one of the best arguments for basic science I've ever heard, right? So this was serendipitous discovery of people who love pond scum making something that has revolutionized neuroscience. In the last 3 years, Carl Dieseroth at Stanford, who's a big advocate of this, has had 15 Science or Nature papers in 3 years. Every other month, it's like anxiety explained, depression explained, addiction explained, and it's kind of crazy, but it's amazing. So why is it a big deal to put these light-sensitive channels in specific places and turn them on? Well, it's specific. This is the main thing, right? So, genes are a big part of what determines what your tissues become, right? And so you can have 2 neurons in the brain 10 microns apart. One of them expresses one set of genes, and this one expresses a different set of genes because they're part of different circuits. And the genes have helped determine what circuit they're a part of. So you shine blue light everywhere, but only the one that's genetically modified by you, because it has those genes, gets switched on. The one 5 microns away doesn't. This is amazing, right? This is astounding. Because if with electricity, you turn on everything, right? And that's the big problem with things like deep brain implants for people with epilepsy and Parkinson, these kind of sort of motor tremor, right? Is that you turn on everything in the area, and people become like, there's a joke that people become like teenagers again. It turns on lots of different circuits, and so you get sweaty and tired and emotional, and you just become all over the place because you're turning on lots of circuits that you don't mean to hit, right? And you can use a channel rhodopsin to turn on neurons. You can use halo rhodopsin where you shine yellow light and it pumps negative ions into the neuron and hyperpolarizes it, and then it won't fire. And so one of the sensational first experiments of some of this is a mouse expressing channel rhodopsin in the motor cortex only. It has a cannula with a blue fiber optic going in. When you shine blue light in the mouse's head, it runs in left-handed circles. And that's because they aimed the cannula to the right side of the motor cortex, which makes it turn left. Which is amazing. More creepily, this is a mouse that's expressing channel rhodopsin in hypocretin neurons, which are thought to have to do with arousal, sleep-wake. And so you're shining blue light on a lot of the brain, but only the neurons that express this set of genes are being turned on. So you have 20 hertz pulses of blue light. This is turning on a specific set of neurons, hypocretin neurons. And then if you wait a few seconds, the mouse wakes up. So you can start to causally tease apart the circuitry that's causing sleep-wake transitions, for example. So I'm talking about doing things in mice for this idea of neuromechanics. Should we use mice? What about fruit flies? Or what about worms, where there are optogenetic tools and they're much simpler systems? Well, I think mice are the right system for neuromechanics because they're big enough that we can certainly think about doing perturbations at the level of the body. Maybe even if someone wanted to make a very small exoskeleton for mice, it would be great to be able to perturb single joints. We can do X-ray fluoroscopy, so we can get nice data. This is another very interesting challenge for computer vision, actually. And the biologists working on this are getting somewhere, but I think it would be a real opportunity. So you can do X-ray movies, right? So you can take slow-motion videos of skeletons of intact, freely behaving animals. And that's what this is. So this is a high-speed video of a walking mouse, and you can see its skeleton. It's a bad one. You can do much better quality than this. I did this quickly. But, it's an interesting problem of extracting meaning from this. And it to my eye, as always, it doesn't seem that bad. But then, of course, you try to code it, and you're like, oh, this is really nasty. So expertise brought to bear on this would be really cool, especially because things like this are happening. So you can do biplanar methods where you take 2 views, and then you CT scan the bones of the animal, and then you fit the bones to the X-ray data. So you have that animal's actual bone structure mapped onto the 3D coordinates of where it was in space while it was doing stuff. So this is a mini pig chewing. Mini pigs exist, apparently. And this one is chewing. And you can implant beads if you need to on the skeleton and track those beads. But a lot of it is still done by what they call scientific rotoscoping. And as my understanding of what that means is you have these 2 views, and you look at them on your monitor, and you position the CT scan bones over the X-ray so that it looks right. And you do that 1 frame at a time. That's not good enough. And it's got to be, with beads, I think it should be fine, right? But I think you'd like to do it without having to surgically implant beads on the bones of these animals, right, as well. So that's a really interesting problem, and that's going to get really big. The fluoroscopy is getting quite big because markers have all these artifacts, right? It's a huge pain. If you put markers on the skin, they flop around. You'd really like to know where the bones are and what they're doing, and this gives you that. And for mice, if you put markers on mice, I don't think they'll last long. You can make them not delicious, but they're still going to chew them off, right? And for mice, you can leverage these big literatures. So there are big literatures on human and cat neuromechanics that we can leverage in a mouse if we use mice. So I'm talking about the argument for doing this in mice. What about the genetics of it? So our ability to use optogenetics depends on targeting specific tissues. Well, actually, of course, mice are a genetic model system. And so if we think about what we can do, well, we could certainly make mice where we can turn on muscles specifically without turning on sensors. So if you use electricity to turn on muscles, you might also hit other stuff in the nerve cord next to it. And in the time we wrote this grant, mice, you can buy mice now from Jackson Labs, where if you shine blue light on their motor nerves, it turns on muscles. And so we started doing experiments with those mice. What about turning off sensory input? Well, we're getting there. We know a lot about what genes are on in your sense organs, but not in your muscles. And so we can do that. What about getting at the spinal cord? That's promising too. We know a lot about the genetic determinants of the circuitry in the spinal cord, although it's, of course, early days. So we're working on this. Yeah, that's going to get better. So right now, if this is the spinal cord of a mouse, we know that certain genes are expressed in different populations of neuron up the spinal cord. We have hypotheses for how they're hooked up and what they do to the gain of the mouse, right? So this is, but it's a frontier. So the models of how these things are linked right now aren't mathematical yet. They're not quantitative and predictive, but they're close. They're arrows on diagrams, like this, right? So there are big pools of V2A neurons, and they inhibit the V0 neurons, and then they go across the spinal cord to the other side, and they talk to these neurons, and these turn on the left leg, and these turn on the right leg, and that's the circuitry that produces trotting. And so with optogenetics, you'd like to be able to say, okay, well now I can shine my yellow light in the spinal cord and turn these off. But we don't quite have the mathematical models that let us say, when you do that, I expect the left leg to lag behind the right leg by a third of a cycle. And that's what we want. Or even what's going to happen also is that you're going to say, actually the model says you turn that off, nothing's going to happen unless the mouse is perturbed. Then it won't be able to recover. And we'd like to be able to test that as well. So we're not quite to this level yet, where we have these sort of mathematical models for the spinal cord. They're getting there. So that's the neural side of things. What about the mechanical side of things? So we're working on this now. This is really fun. So we'd like to have a treadmill-based system where we can have closed-loop experimental design. So we have a freely running mouse on a treadmill, and we can wait for it to be in a certain state and then trigger the neural or mechanical perturbations. So you'd like to be able to blip on and off parts of the spinal cord or the sensory system or the brain while the animal's running precisely based on how fast it's going, how long it's been going that speed. You might like to apply small earthquakes to see how it can recover from those earthquakes, right? And so we're doing this, actually, and it's going pretty well, but that's a big part of why we're here. And you can imagine extending this. So this is an earlier diagram, but you can imagine turning off sense organs here. You can imagine trying to get at the spinal cord to turn things off, and then lots of work is already done up in the brain. And this is pretty amazing because you could see that you might have access to the whole hierarchy. You can work from individual sensors in the leg through to the neurons in the spinal cord. They're doing integration and sending muscle commands out all the way up to the brain where you might be doing these high-level computations. And so what we're working with now is a simple feedback-controlled treadmill system where we have mice running on a treadmill. This is one of our rough terrain experiments. And we're tracking the animal in real time with OpenCV on an Linux box. And so this is a pretty simple algorithm. We find the dark blob, we fit a contour to it, we fit an oval to that. We use a simple Kalman filter to smooth the data and get the position and velocity of the mouse out. And then what we do is a little cute. We have a position reference, and which is the middle of the belt, and a velocity reference, which is 0 speed in the lab frame. And we close the loop with the treadmill belt speed. So when the mouse runs up here, the treadmill belt speeds up. When the mouse slows down, the treadmill belt slows down. And so it isn't closed super tightly, but what you can see is that the animal freely runs on the system. And the cool thing is we have this real-time feed of what it's doing, right? So we can just wait until it does 10 strides at constant speed and then say, okay, turn off this part of the brain, or okay, apply this earthquake, right? And in this experiment, we were just doing how do they handle rough terrain, because that's been an interesting thread for our research for a while. And so here's what it looks like just from the side. So they're blazing away. They love to run, actually. Mice will run kilometers. You can get strains of mice that run kilometers in a day. And you see that when it decides to stop, the system slows down and we don't shove them into the wall. So no one's touching anything here, which is great. This was our best mouse. They vary a lot by individual. There you go. And so we have a couple cameras hooked up. One of them is a simple firewire camera that's just doing the tracking, and then we have a high-speed camera. So this is our tracker computer, and then it's synced up to another computer that runs a high-speed camera. And this is really cool, actually. So when it does a good behavior, say running at constant speed for 10 strides, it sends a packet over to the other computer, which then stores high-speed frames. And so it'll say that segment of data was good, grab it. And so we're using this closed-loop triggering just now to record data. And so we get these videos. So this is, and we have mirrors set up on either side, right? So there's a 45-degree mirror here and a 45-degree mirror here. So you get the top-down view. This is 280 frames a second, grayscale. And then you get the view of the right side of the animal and the view of the left side of the animal from the mirrors, right? And the idea with the rough terrain is to perturb the gait and see what the structure of the controller is for the legs. And we can talk about that more. So we would love to get an automatic estimate of the posture of the animal for each frame from this video. That would be amazing. And it would actually be a pretty big deal, I think, for the medical community. There are automated gait analysis systems for mice, but they tend to use a transparent treadmill belt and film from below. And all they get is paw contact times. They can't see the paw when it's in the air, so all they get is just this pink, oh, the paw's on the ground now, and then, oh, the paw just came off. And that's useful, but it's not as good as we need for time-based models that will actually be relatable to equations, which is important. So we have a closed-loop system, and then the last thing to talk about is some of the theory we're trying to put together for this. So there's a large literature of neural circuitry, obviously. So we have lots of models of how neural circuitry produces things, although for the spinal cord it's complex, it's not quite there yet. We have these simple models at the level of the whole animal. So we talked about pogo sticks and different sort of mechanical models of how the animal's moving. But we'd like something in the middle, actually. So a big mapping. So these give you leg phases. They tell you, what the bursts of oscillations are in the legs. These tell you what the overall body behaves at. But a big component that's missing is how the legs are timed relative to each other and how they converge to link these two things. And so we've been working on this, partly using dog data. So this is Norman. This is my postdoc's landlady's dog, Norman, on our horse treadmill. So we've kitted Norman up with a bunch of markers, and we want to estimate the gait and during gaits and gait transitions to make a good model of it. And so these are the paw markers. We can take those out. So this is laying the foundation for why we want to extract these things in mice, right? What we want to automatically track those videos. So what you get out of those paw markers is you can look at them as oscillating, right? They give you roughly sine waves in the fore-aft direction, and then you can turn that into a phase variable. So front left, back left, front right. So these ramps are the legs going through 0 to 2 pi cycles, right? And you can see here it's a trotting gait because the blue and green, front left and back right, these two are in phase, and the cyan and red, front right and back left, are in phase, but they're 180 degrees out from each other. So this is trotting, and then here you get into a walk, which is 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, right? So you can take those 4 leg phase variables and take differences between them. We use this nasty convention here, which makes trot simple, but you can use other ones. So you take that minus that, that minus that, that minus that. You're down to 3 numbers on a 3 torus that look like this. So now you've got trotting. This is 0 stroke 2 pi for these 2 variables, and then pi for this variable, and then coming down to walking, and then going back up to trot. So now you have 3 numbers in R3 that are on a 3 torus, right? And those describe the gait. So, for instance, walking might be here, and trotting might be here, and you might have this cloud of points for what walking looks like in the mouse as it moves. We wanted to model this, and so we came up with a cool way to do it. So what we thought we'd do is, on this 3 torus, we could lay down a potential function that drives the state of the system to certain gaits. right? So we're taking a sort of dynamical systems approach to this. Let's write down equations for a potential v. We can make it a function of speed, so you can have this knob speed that changes from walk to trot. And then you lay this potential function down on this space, and then you make the change in your current state equal to the gradient of the potential. Right? So it's basically like a marble on a mountain, right? And if the ideal walk is here, and you get kicked up here, this potential function will drive you back to the ideal walk. And if you dial the speed up, the minimum that is at the gate moves from walk to trot. So we have a way to model gates. And this is useful. In the biomechanics literature, this stuff tends to be pretty descriptive, right? Muybridge started it off with his high-speed video of horses in 1910. You know, you measured the timings of the footfalls, and you go, okay, you draw these like gate diagrams, and you say this is what the gate is. But that doesn't really lend itself to mathematical quantification, right? And it's hard to model and make models that are predictive. And so Simon, my postdoc, has been working on this, and this has worked out nicely. Another aspect of this we've done is we've tied this potential down to certain points based on stability margins. So this is a bit of an aside, but you can make the potential have minima at gates that are statically stable, that kind of thing. So you can make this a hypothesis about stability. And you have these nice knobs that control the structure of this well. And so the model does pretty well. So blue is model and red is data, and we can use the model to generate a gate transition. So this is the model. We're at low speed, we ramp up the speed, and it goes from walk to trot, which is actually really cool. And we made a prediction with this, actually. So based on this, we can run the model at walking speeds and then inject noise and make a prediction for what will happen to the gate, because the potential has a certain structure. And we did that, and what we found actually is that based on the model findings, if you're at walk and you get noise injected in your legs, your gait doesn't get isotropically noisy. It should move towards trot. And so we came up with a way to test that in animals by walking them on rough terrain. So we thought, let's take some animals, put them on rough terrain, and see what happens. And we did that at first with dogs, with sensors. So we had 5 dogs. We have flat playing fields, and then we have some bumpy, mogully areas out in the English countryside. And working with Alan Wilson and John Lowe from our group, we used our data logger. So we have a GPS unit here, and then IMUs that look like this. And we put GPS here, and then IMU on each leg, and then they're all linked via RF and synchronized. So we get position and speed from the GPS, which is really nice, and then we get 9 DOF information from each of the IMUs. So we get 3-axis accelerometer, 3-axis gyro, 3-axis magnetometer. And then from those, we can estimate those limb phases. And so my undergrad, Michelle, almost killed herself galloping these dogs over rough terrain, which was, I had to tell her to not do too many trials of that because she was going to hurt her. Dogs gallop pretty fast. And the ground is quite, the perturbations are sort of 20 centimeters, right? So, and that was actually super interesting, because what we found is, if this is the ideal walking gait, this red circle, on flat terrain, they did that with a couple points moving up towards trot. And if this is normal trot on flat terrain, the cyan points were the walking gait on the rough terrain. And you don't get this isotropic blurring of the gait. What you get is something that does move up to trot. So that's really interesting. It suggests that the controller for gait in these animals looks like that. And in mice, it gets really interesting, because mice are thought to trot throughout their speed range. Right? So mice trot throughout their speed range, even when they're going slow. No one knows what will happen when we do it on rough terrain. And it'll tell us a lot about the structure of their controller. But we need the data to do it. We've done a similar experiment in horses. I'll just do this quickly. So you can put thousands of markers on horses and put them on our treadmill and get lots of data. We did some neuroclock experiments here. And I'll just say quickly that their gait changes with exercise in interesting ways. So the horses, we had 6 horses, and we walked and trotted them and then walked them again. And the second walk became more pacey, which is really interesting. And of course, it wouldn't be exciting unless we were doing stuff with the robot. So now what we're doing is we have an undergrad from Penn, University of Pennsylvania, from the COD lab, from the GRASP lab at Penn, and he's putting our controller into Gary. And we're going to take Gary outside and run Gary on the same rough terrain that the dogs were on with our controller and see if it does any better. So we can program the robot with a crawling gait that's analogous to the walking gait in the dogs, but then make it so that it steers the gait towards trot if it gets perturbed, and we'll work with that. So I should wrap up so that we have some time for questions. I think that I'll skip some of these future directions. So I think it's more relevant to stick to the challenge today. So why don't I wrap up quickly and say. So, some of the experimental findings, we found that insects and dogs use a different control strategy than humans, despite having similar ones on rigid surfaces. We think that may have to do with stability in humans and energy in the multi-legged animals. Our robot experiments are where we get this notion that we think it may be to do with energy costs. Rewriting the neural code has shown us the context dependence of control. And lastly, our stability-inspired model of quadrupedal gait does a good job of predicting what animals do on rough terrain. I hope that this shows in some ways that locomotion is important in both across biology, medicine, and technology, although I didn't talk a whole lot about medicine. There are new techniques that are extraordinary, and they're really going to lead to rapid progress, both in understanding the structure of animals and in helping to cure disease. And I think that this combination of neuromechanics and optogenetics is really extraordinarily exciting. And you could see a point where we're able to go from ion channels in the spinal cord of the mouse all the way out to the center of mass forces in an animal that's a model system for human disease, which is crazy to think about. So with that, I'll thank you very much for your extended patience and take lots of questions. Yeah.
So, on beginning for the cockroaches on the latex surface.
Yeah.
In a natural environment, do they encounter surfaces that soft?
Great question. They do. So they live in leaf litter. So they run on twigs and leaves and dirt, and they want to be under it and around it all the time. It won't be a clean 11 Newton per meter hooky and elastic surface. But certainly they deal with these kind of things. There's an interesting question there about whether is your perturbation designed to be naturalistic or to probe the control structure. And in that case, we're asking the question, we're asking the question, what do they do on that surface? Can they stiffen their legs? And the answer was no. It's reasonable to think that they would encounter soft surfaces. But I think it might be useful to do anyways in order to probe the control structure. So, yeah. I think there's two reasons to do these things. You might do things that are totally unnatural because you want to probe the control structure, right? But then you also want to make sure at some point that you know what's naturally relevant as well.
If I understand well, so you said that humans stiffen their legs running concussions because they are afraid to fall, right? Mm-hmm. And what do you think about kangaroos? Like, they are of the same height, weight, they have two legs. What's your guess?
So no one's done that experiment, actually. They've done lots of work in kangaroos. They're really interesting. I would... Yeah. So they have a very compliant gait. I mean, they're even more specialized than we are. So I'm just wondering if they might not be able to stiffen their legs. So we do it through an extended posture. So we do it by landing with a knee that's more extended, and that stiffens it. And so they could probably do that. So I would guess that they would adapt. They would do something. They wouldn't just ignore it, like maybe like the roach we think does. But they may not be able to stiffen their leg to compensate. Kangaroos are amazing. They can go. It costs them the same amount of energy per unit time to go 3 meters per second as it does like 8 meters per second. So their cost of transport doesn't depend on how fast they go, and it's thought to be the giant tendons in the hopping. So they store and return so much energy in those tendons that it doesn't cost them to go faster. And so then you ask the question, why doesn't the kangaroo always go its maximum speed? And the answer is it's risky. Yeah.
Back to the cockroach.
Yeah.
Does the cockroach still run at its optimal speed when it's on the soft surface, though? So is there a reason for it to stiffen its legs?
It wasn't. They didn't. They blast right across it. The speed was the same. So, yeah,
It wouldn't need to stiffen its legs.
it doesn't need to stiffen the legs. I think it would care. You know, so the argument with them is, if they spin out of control, they get eaten by a primate like us. Right. So the argument for that stability is important, is that there are lots of primates in the forest that see a delicious Glabrus discoidalis cockroach and go, and if they spin out of control, they're going to get eaten before they get into the leaf litter. So, and I spy that mostly.
So they have less stability when they're on the soft.
I think so, yeah.
Yeah.
But we would love to know that. We would love to say that's where we would love to have the cannon and the elastic surface, which is hard. But in the mouse, you can imagine having, you'd like to perturb them and have them either turn off parts of the brain or the sensors, or have them on rough terrain and then perturb them.
Now in the experiments, you're assuming that there is gravity. Have you ever thought about the absence of gravity and to see how they behave?
That is super interesting, and this is going to be a letdown because I don't remember the detailed question. But so Jim Usherwood from our lab is doing that in mice. So he has a grant from the Wellcome Foundation to raise mice in a centrifuge. So they're building a giant centrifuge that can handle mouse cages, and there will be mice that spend their whole lives in hypergravity, I guess, right? It won't be hypogravity, it'll be stronger gravity. And I think the questions center around, does the nervous system or does the musculoskeletal system re-optimize itself in terms of energy consumption under those conditions? So you can make simple biomechanical predictions about what gait will minimize energy consumption under higher gravity. And the question is, do mice grow up, under those conditions, do mice grow up optimized for the new gravity, or are they constrained by their genes for normal gravity? Turned out it's been a hard project to get going, because if you have it's got to be like a 2 meter, 3 meter more, it might be a 5 meter centrifuge, and it's got to run all the time, and it's got to be instrumented, and I'm glad I'm not going to be in that room and/or designing it. But it'll be sweet once it's going. Yeah, great question. What do we know? So there have been cool studies on humans. So we did that actually on that horse treadmill. So what you can do is you put people in a big harness with a really elastic belt pulling up on them. So you can have people on a treadmill and then you reduce their weight, but not their inertia. So you can manipulate both. So you can pull up on them to reduce their weight, but not their inertia. But then you can pull up on them and then also add mass. So you can sort of reduce their weight, but not their inertia. I'm mixing this up, but there are ways to manipulate these things, and people will re-optimize for these things. So people do, I think, find the new gait that is the energy minimum. So in general, that's a big hypothesis. So one of those theories of control, optimal feedback control theory, says that the nervous system optimizes motor inputs to meet some cost function that is a mixture of completing the task and using the minimum energy. And it can do that. It can handle gravity changes, but there may be things it can't handle. Yeah.
So you showed us a few vision tracking things that you had implemented and used, like the sorry, was it a cockroach? You had 6 tracks. So what's the challenge? You said you didn't know, or you didn't have an algorithm to track the mouse on the treadmill.
Yep. So we would like to get, let's see, where is that going to be? We would like the paw coordinates from those slow motion videos. So I think she included it in here, she may not have. So we've been manually clicking points to extract the position of the paws. Good. I was hoping to hear those. To extract the position of the paws when the mice are running on the system. So let's see. I hope she included that in here. And we would love to automate that. So what we would like is, here we go. Where did that go? There you go. So here's our ultra crude setup right now. So we have the high-speed video, and the students click the pause in each frame of the video. And we would like the automatic estimation. You can use the mirror views, you can use the top-down view. We would like the 2D coordinates of the paw in each frame automatically tracked through the movie. And it's a bit nasty. There's shadows. The paws change shape throughout the stride, right? Yeah. However it happens, I would like the positions, the 2D coordinates of all 4 paws for each frame throughout a movie. And the movies, to remind you, look like this. I have it. So here's a nice one on plain terrain. It's a little dark, but let's find one of my new ones. And with these, we can do all kinds of things, right? So this is ready to be tied into models going into the circuitry in the spinal cord. So yeah, so we'd like to, out of that, you can see a little white paw going. It's going to be harder on the rough terrain. I'd be very happy on the flat terrain. Rough terrain would be amazing. And that's what we want out. So one idea is, if we want to get into ideas, one idea is a phase-based model, right? So the paw, because the paw changes appearance as it goes through each cycle, but it probably looks similar at each phase. So like, when it's at the front of the cycle, it looks one way, and when it's at the back of the cycle, it looks another way. A phase model would probably work well. So maybe something template-based that, I know templates are terrible, right? I've heard they're terrible.
Oh, yeah.
If you had something template-based, but that was phase-dependent, it could work well. So if the system was keeping track of the phase of the cycle of oscillation and knew what the paw looks like at each phase, that could work. And then it could predict what it should look like in the next frame based on the estimate of phase. I bet that would work. But it may be that that's stupid and narrow-minded, and actually be better to do something more global that takes into account the body. You can do things like, one approach I was thinking was, you can get the body easily from this view, and then you can shift your coordinate system to extract the side views. And so you have a body coordinate movie of the side views. And then the paws oscillate in relatively fixed cycles relative to that in that coordinate system.
Have you got a 3D model of the landscape?
No. I'd love
Drawing
one.
back on top of that.
Sweet. Is that hard to do? Cool. We don't have calibration objects, so that's toughy. We didn't put calibration objects into this. We could. That was a big deal. So that's what we like, 2D paw coordinates for all 4 paws throughout the movie.
Why 2D?
Actually, good question. Actually, just 1D would be fine, but I think you're going to end up with 2D, right? I don't know how you would.
I mean 3D.
Oh yeah. Oh, 3D would be great. Great. For our phase models, we only need 2D, but 3D would be great. Thank you. Yeah. That's going to get really interesting when we start doing perturbations. If we can't do the cannon in the mouse, but we might slide the ground out from underneath the mouse. And when we do that, we'd love to know where they put their paws.